Hello, everyone. As a reminder, please submit your questions to the to our panels because Dr. John Mazziotta will entertain and provide the questions to the panelists. At this point, it is my pleasure to introduce Dr. John Mazziotta, Vice Chancellor of UCLA Health Sciences and CEO of UCLA Health. And John, as moderator of the Amgen R&D Academic Strategies panel, I welcome you to begin your panel and introduce your panelists.
Well, thank you, Mark. Welcome, everyone. A good audience and great panelists today. So I'm happy to be the moderator to help guide everyone through an interesting discussion about academia and R&D and the cross-links between those two entities. Focus on Amgen, obviously one of the great and largest biotech companies in the world, and right here in Southern California for 41 years. And today we have a group of distinguished panelists from Amgen, from R&D and also from the development medicinal chemistry side. And so what we're going to do is go around and ask each panelist to introduce themselves and to provide, a specific comment on an exciting activity that's happening right now in Amgen or some other aspect of their work that they think would be relevant to this panel, talking about the interface between academia and biotech and pharma developments. So let me start with Ray Deshaies from Amgen.
Hi, John. Thank you very much for the introduction. So I'm currently the head of research at Amgen, and there's a lot of things I'm excited about right now. Just to name one, for example, we have a lot going on with our bispecific T-cell engager molecules. Just this past fall, we showed that this mechanism of recruiting T-cells to solid tumor cells showed promising initial data in early stage clinical trials in both small cell lung cancer or non-small cell lung cancer as well as in prostate cancer. And so, that's an example of really bringing forth a new modality that has the potential to really have dramatic results in therapy. Another example much earlier is that we're building something that I helped spearhead the creation of called the Induced Proximity Platform, which takes that idea of BiTE molecules, of bringing two things together to enable biology to happen, and it generalizes it into any kind of molecular construct that could bring two things together. And some of the examples we're working on involve targeted protein degradation, where you bring a member of what's known as the ubiquitin system into proximity with a target protein so that the ubiquitin system can tag the target protein for elimination from the cell. So lots of exciting stuff going on. Those would just be two examples on top of mind right now.
And maybe you can just say how long you've been at Amgen.
Yeah, sure. I've just been at Amgen for, I think, my 4th anniversary, if all the LinkedIn messages I've been getting are correct, just passed maybe about 2 weeks ago or so. Before that, I spent my entire academic career at Caltech, where I was on the faculty of the biology department. So, I made a relatively late career transition from being a basic researcher into working in biopharmaceuticals.
Great, thanks so much. Let's move on to Philip Tagari.
Hey, John, thanks so much for the invitation to speak to this group today. Unlike Ray, I've been at Amgen for 23 years this year. And then prior to that, over a decade at Merck. So a real industry veteran. And see that in our space, there's probably more innovation occurring now than perhaps at any point in the last 20 to 30 years. A couple of things that I think are really top of mind for us First is the real successes of oligonucleotide therapeutics, both our own internal small interfering RNA efforts, as exemplified by some of our clinical programs addressing Lp(a) as a form of high-risk dyslipidemia. But more importantly, the message that has been kind of sent by the astonishingly rapid development of mRNA vaccines. And so this has really kind of brought the whole field of oligonucleotide therapeutics to the front. And then I think the other thing that is really interesting and exciting at the moment is the whole convergence of life science and computational research. And so tremendous opportunities at the interface between biology and computer science.
Great. Well, thank you. Appreciate those comments. And I should say that I've been at UCLA 37 years, so that's more than the two of you combined at Amgen. But we'll see if somebody can beat that record. All right, on we go to Tara Arvidsson.
Hi, John. Thank you so much. This is a great opportunity to be able to speak to this group, and I really appreciate the fact that I've been given the opportunity to represent, in this case, our San Francisco office. I was based in our Thousand Oaks office for about 12 years, and then have been up here in our San Francisco site for the last probably, I think, 4 years now. The things that I'm most excited about, Ray recently or just alluded to the fact that our T-cell engager platform is showing some really promising early activity for patients. And that's just the tip of the iceberg, understanding what we can do with respect to bringing T-cells to tumor cells. But the other element that I'm really excited about is how can we continue to boost that activity? How can we continue to engage the immune system even when the BiTE molecules may not be there in order to spread an immune response so that patients continue to respond over time? And so thinking about how we're going to continue to build on these modalities, I think offers a great opportunity, not just for industry, but also in thinking about how we're going to collaborate more effectively or effectively with academic scientists who are really spearheading these types of basic biology. And to that end, one of the other initiatives that I have the opportunity to work with is this platform that we call Fastlane, where we take really exciting programs and we move them very quickly through our development process so that we can take them to patients as quickly as possible. And so along those lines, the way that we see that Amgen intersecting most effectively with our academic collaborators is that if there are opportunities to synergize, opportunities to bring new modalities, new therapeutics into the industrial setting, we really do focus on trying to make the most of them as quickly as possible.
And maybe I missed it, but what's your role in San Francisco?
So I'm executive director of oncology research, and I oversee the research activities associated with our T-cell engager platform.
Great. Well, we'll come back to that in a minute. So thank you, and let's move on to Chetan Goudar.
Thank you, John. It's great to be here. I come from the process development side of Amgen, and what we do is, we take these wonderful molecules that Ray, Philip, and Tara described to you earlier, and we convert them to medicines so that they can then go to patients and serve the purpose they're designed for. So I've been with Amgen for about 9 years now, and I'm a biochemical engineer by training. The most exciting thing for me is when I look on the research side of Amgen, it's all these novel molecules that are coming our way. So that's incredibly exciting. And if I look on the other end, we have a couple very exciting molecules that are with the agencies for review and hopefully approval soon. Top of mind is sotorasib, our KRAS G12C inhibitor, which we were able to move very fast through the clinic and into patients. And then also we have a molecule called tezepelumab for asthma that's also been submitted for review. So again, I think some exciting developments which are very, very close to patients. and equally exciting are the new molecules that could be coming our way or that are coming our way literally every month or so. So that to me is exciting. And this interplay and interaction amongst the different functions, I think, and the cross-disciplinary nature of the work is what is critical for us to advance our ideas into products for patients. Thank you.
Thank you. And finally, Saptarsi Haldar.
Great. Thank you, John. And again, on behalf of all of us at Amgen, thanks for the opportunity to engage in this great symposium. So I'm a physician scientist. I'm a cardiologist by training, and I'm currently a vice president of research at Amgen based at our South San Francisco site, and I lead all of cardiovascular and metabolism discovery. Before this, I've been at Amgen for 3 years, and before this, I was a faculty running a lab at University of California, San Francisco, and the Gladstone Institutes based at the Mission Bay campus. So what am I excited about? I would say, I would add to this discussion, I'm very excited about the possibilities of leveraging human multi-omic technologies, genetics, proteomics, metabolomics, and single cell based approaches, not just for target discovery. I think we often think about, the first wave of omics for, picking a target and interdicting it. But I think that's just the tip of the iceberg for multi-omic technologies. I think it can fuel the entire drug development pipeline end to end and help us answer questions like who will respond preferentially to a specific therapy or therapeutic axis. And we can really, I know the word is used a lot, but I think we're truly moving into an era of more precision medicine, not just for the ultra rare orphan diseases where there's highly penetrant defects in a single gene, these monogenic disorders, but actually complex diseases and traits where we can actually use these multi-omic technologies to try to narrow this down to be more precise in a manner that will enhance efficacy and safety and ultimately the ability for a drug to help the patients that need it most. And so I would say, that's really something that's evolving. I'm very excited about, and I hope to discuss today.
Great. Well, thank you. And maybe just give us a sense, you've all been at other either academic or corporate institutions before this. What's the difference at Amgen? What attracted you, and what is the excitement there about the development of new therapeutics? What is it that keeps you there, got you there, makes it different?
I'll jump in, John. I think you just heard some of it. The people, there's just an extraordinary group of colleagues to work with. Every day I learn from my colleagues. I'm impressed by their commitment, by the depth of their knowledge, their experience, and their passion for science. So I think that's one very important thing, is just the people. Second is the culture of taking risks, of really thinking aggressively about developing molecules that are really pushing the envelope. So we just put through in December, I forget, it was like 5 or 6 new programs, and all but one of them was either a first in class or a best in class. This is not a Me Too group of people. This is a group of people that is really trying to push the boundaries, invent new medicines, and fundamentally change the treatment of disease. That's just really exciting.
You all agree? Other points of view? Culture.
I'll say, as someone who, like Ray, came from an academic faculty position, within the last, 5 years or so, I always get asked, so what's different, in your new job, and is anything surprising? And the one thing I'd say I was most surprised by how many things are the same actually between academia and biotech and biopharma, and same in a good way, as Ray said, quality of people, quality of science, commitment to a mission. And so obviously, we're in the business of making molecules, and academia is in the general enterprise of knowledge dissemination and training. But I think there's a lot of intersection. We also disseminate knowledge. We're also committed to mentoring and training as well. So much more similar than different.
I mean, the one thing.
Go ahead.
Yeah, the one thing I like to the culture piece, right, that Ray mentioned is, even though we are 20,000+ people today, it does not feel like that. If you ask somebody for help, or if you reach out to somebody, people are very willing to help and work. So I think Amgen has managed to maintain over the 40+ years some of that small company feel when it comes to the ways of working. So I like to tell people, when I joined 9 years back, I came for the science, but now I think I'll stay for the culture, which is both the aspect that Ray talked about, us trying to constantly push the envelope across all dimensions, but just also the way we go about doing our work, I think is pretty unique, and that makes the place very special.
Well, let's stay on that topic. So two of you came directly from academia to Amgen, and it sounds like culturally there's a lot of similarities, and that's made you feel both comfortable and excited about the environment there. So maybe give me a sense of, since we have academics on these panels and in the audience and people from the private sector, how does Amgen view partnerships between academia and biotech or pharma? Is this a situation where you're always looking to see what can be obtained from academia? You just want to hear about the knowledge. You're not looking for actual intellectual property, some balance of those. How do you choose your partners with academia, if that's part of the answer?
Tara, maybe you could tell them about the BITE collaboration with the Tejan laboratory. I think that's an interesting example.
Yeah, so this is something that's been really exciting for us, is to understand at a molecular basis how our molecules are interacting with T cells, with target cells, and how changes in our molecule structure or affinity can affect the biology. And in this particular collaboration that we have with the Tejan group, they have technology that is really cutting edge and has the ability to look at things that we in an industrial setting don't necessarily have the ability to look at. And so it's been a fantastic collaboration between their scientific group and our scientists, trying to understand how these very subtle changes can actually lead to an improved, safe, efficacious therapeutic. And from my perspective, it's been incredibly illuminating for both of our groups.
Yeah, I would also maybe note that these kind of collaborations are most productive when they leverage, things that Amgen is particularly good at, and that is sometimes providing unique molecules and tools. And so we've had some excellent collaborations in things like, measuring function of individual nephrons with scientists at UCLA, where we have provided unique molecules that, modulate ion channels and cell surface receptors. So the best collaborations are really genuine, co-invention of new science.
One thing, John, that academic labs are really good at, excel at, is striking off and asking questions where you don't know what the answer is going to be, and you don't even know how to ask the question half the time. So you're spending a lot of time, like you're in the dark and you're reaching around trying to find something to grab onto. In industry, what industry excels at is when the problem is a little bit better understood, and you know what you're aiming for, and then you have this amazing firepower to go after the objective. And so what's great in collaborations from our point of view is when we can engage a group that can do this blue sky stuff, like the single molecule work that Tara was talking about, that Bob Tijan's lab is doing. Or, you know, we have a collaboration with IPD, the Institute for Protein Design at University of Washington, where they're coming up with all sorts of, you know, fancy ways of designing new proteins that don't exist in nature. And so we can come up with an idea, let's say a new kind of bispecific T-cell engager that is regulated, so it only turns on, say, at a cancer cell. And then they can go and see, okay, they have a system known as Locker, and can we use Locker to create that sort of entity? Maybe, maybe not. But, you know, they have that flexibility and freedom to go off an open-ended discovery like that. And then if they find something, then, you know, we can start digging deeply into that. So, you know, I think the two bring very complementary, distinct and complementary skills to the table. And when done right, could be very powerful.
John, I'll also add something that hasn't been touched upon. We've been talking mainly about scientific collaborations around biology and molecules, but at a structural level, I think together academic institutions and the private sector, particularly the larger biotech companies and biopharma companies, we have to work together to build an ecosystem. And in particular, part of that ecosystem involves small startups and taking technologies out of laboratories and universities, for example, that are investigator initiated, and actually spinning them out to de-risk that bleeding edge technology and getting guidance from both sides on this, and then ultimately partnering with large companies to guide things based on how you're going to take. Seen it done well for oncology and immunology. I think there's a huge gap in some of the other chronic diseases like cardiometabolic disease, chronic kidney disease. We're just seeing it, but we need much more of that. And I know there's terrific investigator-initiated work at the universities that we could all benefit from and benefit our regions and society at large.
So let's go one step even further to the far end of that spectrum, because now have quite large, well-characterized patient populations and really specific details about them. At UC, throughout the 6 medical campuses, there are 16 million patients. So if you wanted to do a very tailored clinical trial with a well-specified, either genetically or phenotypically, subgroup, these large centers, and you know them all, would find those patients for you and do that trial. Is that something that's in the ongoing thinking and machinery of Amgen or in general in the biotech sphere?
Sap, you want to feel? Sap's the only physician in our Amgen group here, so I'll leave him to address that question.
I would say, the precedent for that has been, I would say, spearheaded in precision oncology, as you know, where we often have availability of molecular characterization of tumors, and also in rare diseases and orphan diseases that are monogenetic, where you often have a genetically defined disease and a rare disease type therapy that will address it. So that's sort of happening. That happens through academic institutions with cohorts or societies, for example, for these rare diseases. But I think there's a huge opportunity to start to unravel the more complex heterogeneous diseases, heart failure, atherosclerotic cardiovascular disease, chronic kidney disease, non-alcoholic steatohepatitis and chronic liver disease, neurodegenerative diseases, the big ticket broad diseases that affect the population. What that's going to require is scale and a deep molecular characterization and phenotypic characterization of patients. And obviously, at Amgen, we've been a pioneer in that area because we've been working with Decode Genetics, and acquired them many years ago, and have a partnership with Decode to do this on a large scale. But I think what we need is to expand that and not just go into cross-sectional population studies, but start to look at specific diseases and start looking at prospective cohorts of defined diseases and trying to really understand their progression and not only incidence, and characterizing them through multi-omic technologies, as I mentioned before, as a start with more, I would say, refined phenotyping, not just ICD-9 based codes, but imaging based phenotyping, for example. And so I think that's really one place where partnering with large health systems will be very fruitful.
So John, just to add to that a little bit, so historically our engagement with Decode has been to discover targets and validate them. And we're now starting to pivot in a direction of using the human omics to identify and stratify patients for clinical trials, identify biomarkers, PD markers. And, you know, that's an effort that's really just getting underway. You may know we did a deal with Intermountain Healthcare. I think it was 2 years ago now. And, you know, we're certainly thinking about other large academic medical systems. So, you know, we may come knocking one day in the not too distant future.
Yeah, I've often wondered if with Decode you could gather phenotypic information from that original group just by going back to them and getting permission and having them enter data on their phones. And you would get some aspects of phenotype, maybe it's behavioral or, a whole host of things that would be pretty passive on the part of the subject and could be done in a highly automated way. Anyway.
Yeah, and we do callbacks. When there's been occasionally very specific questions, they have the capability in the Icelandic population to call people back and do deeper phenotyping.
Yeah. Let's shift gears for a moment before we go
John,
to...
can I just make one other point about diversity in our studies as well? And that's an area that we're very interested in continuing to expand. Diverse populations that we have the opportunity to profile in our studies. And that's where I see that we have an opportunity to work more closely with various academic centers.
Great point.
Yeah, because you have the homogeneous populations pretty well captured genetically in Decode, and also then Intermountain is a much less diverse group than you would find, say, in Southern California. Let's talk about COVID. So all the things that have happened with COVID, how do you see that strategically affecting how drugs are filed, approved, brought to market? How do you think regulators will look at new filings now that it's been shown that fast tracking a lot of these things, not only work, but, saved enormous numbers of lives with a successful mRNA vaccine? How do you think the process will change? Just speculating.
Chetan, you're probably the closest to that.
Sure, yeah. So John, as the world has seen, right? I mean, we have seen vaccines move forward at breakneck pace. We have seen monoclonal antibodies, right, get approved very, very quickly. So there are a couple of dimensions there, right, that come to mind. One is, what have we learned, as members of the biopharmaceutical industry in terms of how we understand, develop, and characterize products? There is a very long list of things there from a learning standpoint that I think collectively the industry will bring to bear on new molecules that we develop. So there will be techniques and strategies there that will help accelerate, some non-COVID molecules also as we move forward. The other bigger piece, which you touched upon, is the agency. How receptive will the agencies be after the COVID experience? And, I do think it's important, right, to make the distinction between the emergency use authorization that was used to have the vaccines approved very quickly. And even now, if you look, Pfizer and Moderna are looking to get full approval now, right? Much after the emergency use authorization. So the emergency use paradigm, I think, will of course be special, and that'll be reserved, I think, to pandemic-like conditions. So we will not be able to leverage some of those things as we move into other therapies. But there are enough things there, like, for example, if you just take comparability, that's a very big part of commercialization where we have to show that as we make improvements to our manufacturing processes, the molecule does not change. And, we had a very traditional view of comparability, which has remained constant for a long time. And now we are learning more about, maybe there is more efficient ways for us to characterize the molecules and maybe shorten the time it takes for us to generate those comparability packages and things of that sort. So I think in the context of outside the EAU, EUA, emergency use authorization, many learnings, both for us developing the medicines and also on the agency side, that I think should help us move quickly moving forward.
So you really think the FDA can and will start to work differently?
Yeah, I mean, as long as we can make, science-based arguments and say, hey, look, here is the scientific basis for us, trying to follow a certain path, I think they will be very open to having those discussions. And we just have to, demonstrate the rigor, but we can do that in different ways now than we used to do before.
Phil, I think it'd be interesting to know what your thoughts are on the implications for the actual discovery of the therapeutic molecule.
So firstly, 41 days, I think, for the Bionext mRNA lead to come from the original sequence of the spike protein. And that was enabled almost entirely by digital technology, okay? So nobody shipped a clone from Wuhan and, PCR'd it out in Berlin and then, FedExed it to Pfizer in Groton, okay? The acceleration of this program was enormously based on digital technology, and the same for the equivalent program in Moderna, where essentially not a single piece of paper was generated from the original sequence transmission through to product in a vial. And so I think this digital acceleration has a profound impact on the pace of development of new therapeutics. And then I would also say, just the ability for industry to collaborate. So you've seen industry partnerships, our own partnership with Eli Lilly for manufacturing of a therapeutic molecule. And so the ability to perhaps partner in key areas in development of therapeutics much earlier, seems a trend that is very positive overall.
Well, I appreciate that. Let me follow up with that with a question from the audience, and which is, what does the first ever approval of an mRNA vaccine mean for oncolytic viral vector modalities?
So as I kind of, broached this topic originally, maybe I'll take that and, Tara can comment. There is an enormous amount of activity in this space. We ourselves have a small collaboration with a startup in Emeryville around delivery of mRNA, potentially as a vaccination strategy. The ability to generate quickly mRNA constructs for personalized cancer vaccines seemed pretty encouraging. Much of the angst around mRNA therapeutics has really been, lack of approval. And we've now seen, the world's single most prescribed drug is an mRNA therapeutic. So I think we're entering a kind of, mini golden age here. And I think oncolytic or oncology personalized vaccines are a real possibility in the next 5 to 10 years.
Great. Thank you for that. Another question from the audience. With bispecific molecules and all their associated advantages on the horizon, what's your view on the fate of monoclonal antibodies?
I can take this. I don't think that monoclonal antibodies are going anywhere. I think that this is all part of one larger repertoire where you have the right tool for the right job. And I think that there's always going to be a need for monoclonal antibodies. There's lots of innovation that's being driven around them now, enhancing various types of effector functions, getting them to do things that we didn't imagine that they would do in the past. So I am not worried that monoclonal antibodies are going to go anywhere. I think that we're just getting to the point where we're figuring out how to make the best of them. And there's going to be a long runway ahead of them.
Yeah, let me put it this way, John. When biologics first came on the scene, starting with Humulin back in the early '80s, you could have said, "Wow, there go small molecules." But two of the top 5 selling drugs in the world right now are Eliquis and Revlimid, which are both small molecules. So, you add to the armamentarium, the old stuff doesn't go away because, it has its niche and its utility where it just does the job better than anything else. So.
Monoclonal's here to stay. All right. That's loud and clear. Another question from the audience. Now that Amgen will be launching TSLP programs for respiratory disorders, does Amgen have a greater interest in other respiratory indications? Those acronyms mean nothing to me, so I'm looking to all of you to see whether they make sense to you.
So maybe I'll start and Ray, you can continue. TSLP, thymic stromal lymphopoietin, which is a master regulator upstream of inflammation, in particular, in both allergic and intrinsic asthma. Our molecule tezepelumab is an extremely potent and selective TSLP antagonist, and has shown in pivotal trials broad, we believe, potentially disease-modifying activity in both allergic and intrinsic asthma, both the eosinophilic and non-eosinophilic. So this is a kind of real breakthrough in the world of asthma. I was fortunate at Merck to be part of the team that developed Montelukast Singulair, so know the importance of asthma therapeutics worldwide. It's still a huge unmet medical need, even with new molecules such as tezepelumab. And we recognize that respiratory disease, COPD, IPF, and just other forms of asthma are increasing, globally as the world's population migrates from the countryside to increasingly kind of polluted and environmentally challenged cities. Asthma is one of those chronic diseases that is significantly increasing in incidence. And so we see there's a lot of opportunity in the respiratory field at the moment.
Yeah, John, talk about an example of monoclonal antibodies still having a lot to contribute. I think tezepelumab fits that bill very, very nicely. Even though it has shown development, and we're keeping our fingers crossed, that just went forward, particularly in the low eosinophilic population. So I think there will be future molecules, and therapeutic inroads into COPD. Aging population, as Philip has noted, worldwide. We have an increasingly urban population worldwide that's exposed to more air pollutants. So, COPD could be an area where, the next big breakthrough is really awaiting us. So we're very interested in diseases of airway inflammation.
Great. So let me combine a couple of questions into one general question for you. And as we're approaching the end of our time allocation, and that has to do with how you attract and engage new talent. So the academic health centers have both graduate students and clinicians who are also MD PhDs. And so the question is, what are the programs? What are the internships? What are the exchanges that Amgen oversees to engage these individuals, get them interested, have a way for you to, test drive some of these people and see if they're a good match for your culture and your interests? And how does that work?
Chetan, PT has used a number of interns over the years.
Sure, yeah, I'll start and then others feel free to chime in. So, John, we have a couple avenues, right, that we use to test drive, if you will. And of course, we also recruit directly as well for full-time positions. But, across the board, across research and process development, we have postdoc programs. And, we actually have had good examples of students from UCLA being a part of those postdoc programs. And I think 8 of 10 times we end up, offering them full-time roles if they choose to, advance their career that way. So that, I think, is an exciting way for them to get involved. And there's a lot of flexibility in terms of the kinds of, work they do and things of that sort. So that's one. We also have internships. These tend to be like, usually over a summer. And that's really meant for someone to get an immersive experience in a very short time, to our laboratories, the kinds of work, the programs we work on, and the kinds of groups that we have, and so on and so forth. So to me, I think those would be 2 avenues that people can use to quickly test drive and experience Amgen. And we're more than happy to, engage students on both of these kinds of activities.
How about in the other direction? Do you ever want to send your research scientists into academics to learn either a technique or to be immersed in some activity that may be easier, cheaper, better as an exchange versus homegrown?
So I did have the opportunity to do something similar along those lines. We were interested, we were pursuing some activity or some opportunities in sickle cell disease. And I did have the ability to go and do basically a summer internship at the University of Michigan, Detroit, shadowing some of the physicians there. Learned a lot, learned it's a very, very tough disease, but it was a really good opportunity.
Great. Well, Mark, I believe we're at the end of our time. Is that correct?
That is spot on. Yes, John. Thank you for the exciting panel.
Thank you all. It's great talking with you, and I'm sure the audience enjoyed the insights and the information that you provided for everyone. So thank you, Amgen, and our panelists in particular.
Thanks, John. Thank you.
Thanks, John.
Thanks, everyone. And please come back at 2:30 for our professor spotlight videos. Those will be airing at that point. Thank you.